I'm Kevin Elmy. In this presentation, we're going to be talking about soil health and using cover crops to help improve the soil health. We farm in east central Saskatchewan. We've been using regenerative agriculture practices, including cover crops. The cover crops we've been using for approximately 10 years, and we've been using regenerative ag principles for just about 20. Hopefully your land does not look anything like this introductory slide. There's a lot more farmers starting to farm the ditches, and there may be a reason why. Because with some of our ag practices, some of our most valuable parts for soil are leaving our fields and ending up in the ditch. So this is hopefully nothing coming from your farm, and hopefully this presentation can help drive your goals in regenerative ag and rebuilding our soils. So what is soil health? What is the big deal? Everywhere you turn today, it seems like has a new article or new research about soil health. Why is it so important? And the next question is, 10 years from now, does it really matter? Is it just one of these fads that are going to show up and people are going to talk about it and slowly fade away? The issue with today's agriculture is, number one, we're dealing with some fairly tight margins. So money is tight. When we're looking at producing these crops, we're dealing with higher fertilizer requirements. Every time we turn around, in order to get the same crop that we had last year, we're going to have to increase it by 5 or 10%. We're finding that in our production systems, we're having to add fungicides to our management practice. We need tile drainage, or we have to clean up waterways to get the standing water off the soil surface, so we can farm these acres properly. All of these problems are just they're symptoms of what the problem is in our production system. I love this quote. We cannot solve our problems with the same thinking that we used when we created them. This is where we're at today. What is the big deal with soil health? Our soils have been changing with our high-tech management, not for the better. So our synthetic inputs, our over-tilling, lack of plant diversity, lack of green plants growing throughout the growing season, removal of animals off the land, and overgrazing all lead to this loss of soil health. We're putting too much stress on our system. So what is this big deal? So if those are our issues, then logically, if we did the opposite, that would start to repair our system. And this is where whether called regenerative ag, I've seen so many different titles and names for this. Once again, new in quotes, the production system, that it doesn't really matter. But basically, what we're trying to do is allowing that soil to heal, build soil biology back into it, and get off the input carousel that we're on. When we are looking at principles, people are saying, okay, where do we start? And when you're talking to or listening to different soil health practitioners, some of them will be talking about four main principles, some five, some six, and I've even had one person up to 10. In the end of the day, there'll be overlap on them. I like using five because I have five fingers. But basically, number one, we need to increase the time green plants are growing in our soils. Number two, we need to reduce the amount of tillage that we're doing. Number three, we need to reduce the use of synthetics. We need to increase plant diversity. And ideally, we should have livestock integration on our land. Those are the five principles that we'll kind of talk about in this presentation. So the first one, which I feel is probably the most important, is increasing the amount of time we have green plants growing in our soils. The importance of that is because a vegetative plant will release up to 80% of the carbon it captures through photosynthesis and release that carbon into the soil. That whole process, the thought process is that that carbon is going in back into the soil. It is feeding that biology, which then is going to go out and it's going to work for that seedling to provide all of the nutrients that we'll need in the future. That is the reason why the plant will release that carbon back into that soil. The other really neat thing is a green plant will tie up free nitrate in the soil. That free nitrate, the a plant cannot control well how much nitrate it takes up. It can control the ammonium, the amino acids, the other forms of nitrogen, but because it's a negative ion in the soil, it just flushes in with the water. What this green plant will do is it will take up so much of that nitrate and then re-release it back into the soil as root exudates as amino acids, ammonium, other nitrogen containing compounds back into the soil, which then your cash crop or the other plants around it are able to use. Tying up that free nitrate is really important, which we will cover later in this presentation. That green plant will also take up other nutrients, so your phosphates, your potassiums, and it's catch and release. That's one of the phrases I heard in one of the presentations. I forget who said it, but it's an awesome way to describe what we're doing. So this vegetative plant's going to take up these nutrients. It's going to re-release it back into the soil. So we're not concerned about having a nutrient deficiency because we have this cover crop growing. The other thing about a green plant is it will support your mycorrhizae, assuming it's one of the mycorrhizal hosts. With the mycorrhizae, when it infects right behind the root cap, when that root is growing through the soil, that will be active in active tissue. It'll go out and it'll do its thing. It'll help that plant grow and get the nutrients and water. But as that root keeps pushing through, that mycorrhizae gets moved, not moved because it's in the cell, but as the root moves forward, that mycorrhizae then the tissue that it's in, that tissue becomes less active, so it has that root, in order to continue picking up nutrients in the soil, it needs to be reinfected with new mycorrhizae in that active tissue right behind that root cap. So we need to have that living root to keep supporting and building the mycorrhizae fungi in our soil. At the end of the day, that green plant will increase the carbon in your soil. Reducing tillage. In my mind, what we're doing does not have to be completely zero till, but what we want to do is reduce the frequency and intensity of the tillage we do. That frequency, so if we are working that soil 4 times in 1 year, can we go back to 3? If we're working 3 times in 1 year, can we go back to 2? If we're working 2, can we go back to 1? So that's the frequency part. So, okay, how do we reduce that? The intensity part is how much tillage are we doing in our A horizon? So that intensity. So if we're dealing with a topsoil that's 30 centimeters deep, and we're working 2 centimeters deep into that profile, percentage-wise, we haven't really destroyed that whole profile, so that soil will recover relatively quickly. If we have 5 centimeters of topsoil and we're working 10 centimeters deep, we've completely destroyed that whole A horizon, and that whole A horizon then has to be completely rebuilt. So it's going to be the intensity is a combination of the depth and how close together are your shanks or your working implement, so that how much disturbance are we doing in that whole soil profile. Nicole Masters, one of the things that she mentioned in one of the talks that I listened to was, for every sin you do against the soil, to make 2 reparations. What that means is, when you go in and we decide we have to do some tillage because we have ruts, because we have weeds, because we have whatever issue we have, okay, that's that's fine, as long as, once again, we're not completely destroying that whole A horizon, the rooting zone. But what are we doing to help repair that? Are we putting down some fish? Are we putting down some humates? Are we seeding a cover crop? Are we going to be seeding that cover crop and then do some grazing on it? So that tillage is the negative. What other two things are we going to do to help that soil recover from that? In the soil health and the carbon sequestration world, reducing tillage does not increase carbon, but reduces the carbon losses from the soil. The reducing of the use of synthetics. You have to earn the right to reduce your inputs. That's one of the messages that John Kempf, in one of his books, one of his chapters, he talks about is you have to earn the right to reduce your inputs. And that means you're going to have to be forward thinking of, okay, so what are my issues in my soils? So if it is low nitrogen, okay, we're going to have to get nitrogen back into that soil system, and we need to have some legumes in with our polycrops. We're going to have to do some fall grazing with some animals to get some urine and manure back on that soil. If it's disease or weed pressure, the weed pressure, there, there's a real great book that we'll talk about later, When Weeds Talk. And basically, when we have that weed pressure, that usually is a combination of low calcium flux in our soil and excess nitrates. So in this case, the weed pressure, okay, we have to tie up those nitrates. So how do we get around that? If we have low phosphate, really easy way. If you listen to Dr. Lane Ingham, she says that in our soils, assuming that we haven't eroded the base nutrition away, we have about 1,500 years worth of nutrition in our soil. When we have nutrient deficiencies, that means we have a microbial deficiency. In this case, with low phosphate, build our mycorrhizae fungi, and our phosphate levels will increase in our availability. The reason is because with the mycorrhizae fungi, they associate with a phosphate solubilizing bacteria, and so that relationship will get more and more phosphate into those plants. If we're seeing disease pressure, basically let's start cranking up those monocrops, because when we're dealing with monocultures, that is where we start seeing issues with some of these disease situations. So by reducing synthetics, this will reduce the carbon losses from our soil. It and that those losses are basically because we are affecting our soil microbiology. And when we start building our soil microbiology, then we have the opportunity to start putting carbon back into that soil. Soil health, the increasing plant diversity. What I work on is a couple triangles, and we'll talk about that in a little bit. And if you've heard me talk before, you'll know about my triangles. I talk about functional plant groups. So when we want to increase plant diversity, we're dealing with our grasses, we're dealing with our legumes, we're dealing with the broadleafs. Within those broadleafs, I break those down into brassicas, non-brassicas, and forbs. So those are our main functional groups. To add the diversity within those, we can deal with warm season and cool season species of each of those. We can deal with annuals, biennials, and perennials. We don't necessarily have to have those growing all at one time, but anytime we can increase the frequency of those functional plant groups showing up in our rotation, that's going to continue to stimulate the different species of microbes in our soil. So adding diversity, plant diversity, will stimulate more soil biology, which gets more carbon back into our soil. And so the last principle that I use is livestock integration. Livestock integration is not necessary, but it sure does speed up that process of soil building. For myself, I do not own animals. I do not want to own animals. I have no experience with animals. Meanwhile, the guy down the road, he is looking for feed. He's looking for pasture. He knows animals. He knows how to manage them. What a perfect combination. So I am growing plants for him to then come over and either graze or hay we do a custom grazing arrangement in this case. So in this case, to get the livestock on your land, there's a bunch of different opportunities you can get into. Management wise, we can deal with a stockpile graze. We can deal with bale feeding in the field. Some people call it bale grazing. The residue grazing. So after harvest, we can have stuff out in the field so they can be grazing, or we can do a full season cover crop that we can either rotationally graze, we can hay and then graze. We have some options there. So the livestock, the grazing part, it helps put more carbon into the soil when properly managed. When we see a pasture like this pasture is not functioning properly. It's overgrazed, it's underrested. We have some issues. The carbon that is going into that soil is less than optimal, and this is where people will say, well, no, I'm kind of doing regenerative ag because we've been rotationally grazing. When you have a pasture that looks like this, the carbon going into the soil is marginal. It's a marginal increase, if any. It's once again maybe protected the losses, but the soil build is not happening because when you listen to someone like Alan Savory and the whole idea of having that full recovery of that plant, this overgrazing part of it, it's not building soil. Alan Savory, he basically says, allow that plant to go through its life cycle and graze it down. Getting into that management system is extremely difficult because how do you start when you don't when you have marginal pasture marginal pasture supply? And this is where the cover crops will then come in. So now we can take some of our cropland, rest that pasture better so that we can get more production. That whole system will fall into place, but to get it started it can be challenging. Now we're going to get into a little bit more of the biology part. The nutrient cycles. If the fertilizer system worked well, we put the fertilizer in the ground, 100% of it would then go from the fertilizer, dissolve and straight into the plant, and there we got plant nutrition fixed. In reality, when you start looking at fertilizer use efficiencies, we are dealing with a diminishing rate of return on our fertilizers. Our nitrogen, at best, we're getting 50%. When we're dealing with our phosphate, we're getting 15 to 20% utilization. What's happening? So there's a few things that come into play. And when we have plant nutrition in our soil, most of it has to go through the soil health, the soil health, the soil biology part. So when we have something in the soil, whether it's organic matter, whether it's manure, whether it's chemical fertilizer, we have our primary consumers. So we have our bacteria and our fungi. The bacteria, they will take the more easily broken down, simple type of nutrients in the soil, break them down and then process them. The fungi, they're slower growing. They will take the more complex plant material, plant nutrition in the soil, and absorb it, break it down, and get it into more of a usable form. If that was as simple as that, of we have the bacteria and the fungi, that's the end of the story. What fun would that be? But basically, that's like going into stocks and bonds. We're investing the money. How do we get them out? We have to get them out somehow. And this is where we need these primary predators. So the ciliates, the flagellates, the amoeba, nematodes. And now with the research coming out, where do the viruses fit into this whole food web? As we begin to understand the bacteria and the fungi, why they're important. Some of the flagellates, the ciliates, the amoeba, the nematodes. Okay, we need those too. Where do these viruses now come in? Because they're even smaller than using the bacteria, the fungi. So we're just starting to crack that case. So with those primary predators, they will eat the bacteria and the fungi and poop them out. when they poop them out, now we have plant nutrition starting to happen. Now we're going to start getting earthworms. Now we're going to start getting that leverage to really start building nutrient cycles in our system. In our soils today, our ag soils where we're growing our cash crops, most on average are extremely dominated by bacteria. So they're bacterial dominated soils. Those bacterial dominated soils are addicted to inputs. As soon as we start cutting off inputs on those bacterial dominated soils, we start seeing production problems. And that's part of the thought process for organic production, where they do a 3-year conversion process from going conventional into organic. The whole thought process is that we can, during those 3 years, you're going to be, cutting off of your conventional inputs and converting that soil and hopefully getting it to be more fungal dominated, so that that plant and that whole system is going to work properly. That's a thought process. Not always happens that way. When we look at our soil formation over time, basically we start with rock. We get simple organisms start growing on them. We start the soil building process. Then we have what they call young soils, so they're more dominated by your bacteria. Then that soil will become more and more fungal dominated, and that's where we produce our trees. And so this is where, when we're looking at agriculture, it's always a battle to keep our soils in that young stage so they stay productive. Another demonstration or example of how this slide will continue going, where we have, on the left hand side, we have bacterial dominated soils, so we have our annual weeds. Then as we get more fungi, we get our more perennial weeds, grasses, then we get into our shrubs, and we get into our trees. As we go to the right, we become more fungal dominated. As we go to the left, we get more bacterial dominated, and we get or more production of weeds. As we start getting more fungi, we get less weeds, because when we start looking at the type of nitrogen in the system, bacterial dominated will produce nitrates, fungal dominated will produce more ammonium. And so this is where, in agriculture, we are always resetting this soil evolution, from going from bacterial to dominated. But most of the time we hit that reset button too hard, so that we keep going back way too far to the left, where we get into the annual weed production. Another way of looking at it is, once again, if you've heard me do presentations, this little slide chart is wonderful, because we can see that on the bottom left, where we're dealing with really early soils, highly bacterial. Then we start to, as that soil starts aging, we start getting more fungal production. So the F to B ratio, that's your fungal to bacteria ratio. So as we get into the fungal bacteria ratio, and this is pound for pound in the soil, at 0.1, this is where we see our weeds. This is where we see our high nitrates in the soil. This is where we see the lack of oxygen. That lack of oxygen normally is correlated back to lack of calcium flux in your soil. As we get a higher fungal to bacteria ratio, so we're at 0.3, then we see our early grasses start really dominating. We get to 0.75, then our mid grasses, our clovers, our vegetables, our 1-to-1 fungal to bacteria ratio. We get our late successional grasses, we get our row crops, native plants. Then when we get to over that 1-to-1, then we start seeing our shrubs, vines, bushes, more native species, and then we get into our trees. So that if you have grown a crop of a pasture or hay, so we've had it in crop rotation, so we're in that 0.1 to 0.2 fungal to bacteria ratio. So we have our weeds, we have all of the issues of modern agriculture. We seed down a forage stand, so that first year with that nurse crop, we grow that nurse crop for weed control, so that we have something growing. We then harvest that first year's crop of weeds and our nurse crop that we have, In year two, you may see some of those annual weeds be in some areas, but they basically started disappearing. In year three, there's no annual weeds. And then what'll happen is over the years and management, then we'll start seeing rose bushes starting to show up. We see silver willows. We see some of those shrubs showing up. So these are all indications that where we are in this fungal to bacteria ratio. So that we start at point one, in year two, maybe we get to point three, maybe to point four, to point five. A plant like Sicer milk vetch, when you seed it that first year in with your perennials, you print Sicer milk vetch you won't see. Year two, you maybe start seeing it. Year three, okay, yeah, it's starting to show up. Year four, bang, it starts showing up everywhere. And with the Sicer milk vetch, it wants to be in that point 0.5 to point 0.6 fungal to bacteria ratio. So when we're at point one, it doesn't have the signal to grow. When it's at point 0.6, it's happy. It has enough fungal component in the soil active that it's going to be able to really succeed. When we start seeing the shrub show up, that now tells us we have crossed that 1-to-1 fungal to bacteria ratio. Now we are 2-to-1, maybe 3-to-1 fungal to bacteria ratio. Now the dangerous thing with these ratios, the fungal bacteria ratio, is they're not absolute numbers. So that our fungal numbers can be quite low, but our ratio is quite high. So that means our bacterial levels are that much lower. When we have these low producing pastures with shrubs starting to encroach, that tells us we're over that 2-to-1 fungal to bacteria ratio. And one of the really interesting things in John Kemp's book is he interviewed a Dijkstra. And one of the things that he suggests to his customers, his clients that he works with, is adding 1 pound, or sorry, 5 pounds of sugar per acre per week in these areas that have low productivity. The thought process would be you put sugars into the ground, it's a simple carbon source. Those bacteria numbers are then going to start increasing. It's going to start breaking down that sugar, increase in numbers. Now the predators have something to eat, so now you're going to start building up your nutrient cycle. So by increasing that bacterial number, now we're going to be able to slide down that slide. So instead of being at 2-to-1, increasing the bacterial numbers. So now maybe we can be back to that 0.7, 0.6, and those shrubs will then disappear because the conditions in the soil are not right. When we have that pasture, and so if we decide if we see the shrubs, we see declining production in those fields, what do we normally do? We spray it out, we work it, we fertilize it, we do do what we do in agriculture. Every time we add fertilizer, that will weaken our fungi and it'll increase our bacteria. We do tillage, that will reduce our fungi and it will increase our bacteria. When we spray with herbicide, that'll decline our fungi, maybe or may not increase our bacteria depending on what herbicide and our soils and what kind of biology we have or so on. But anyways, so that when we break it up, we will go from that 2-to-1, and we say if we spray it, we work it, we put down fertilizer, so we go from 2-to-1. Now we're at probably 0.7. So then when we're breaking up a hayfield or pasture, going to oats is kind of a knee-jerk reaction because we can grow good oats, good clean oats on it. So we'll go from that 2-to-1, maybe we're down to 0.7. We grow that oat crop, we take the oats off, we go out and we do tillage, we spray it, we add more fertilizer. Now we've gone from 0.7, now we're down to about 0.4 fungal to bacteria because we're knocking back the fungi and increasing the bacteria. Then when we go back and we grow the oats that second year, when we're at 0.4, we may see the odd wild oat in some of the spots where now the fungal to bacteria ratio is below that 0.3, but it's still fairly clean. We go in that third year, we work it, we spray it, we put down fertilizer. Now we're below that 0.3. Now we start seeing wild oats. Now we start seeing cleavers. Now we start seeing some of those annual weeds. And one of the things that I'm talking to people about is maybe we should change the word weed to an early successional plant, because that is exactly what we're growing. Is we're setting up the conditions for that early successional plants where that fungal bacteria ratio is below that 0.3 for those things to grow. Just a thought. When we're building mycorrhizae, there's this nice little chart here that goes through, and it shows the crop dependency on mycorrhizae colonization. So the plants that are dependent on mycorrhizae are basically your warm season grasses, your legumes minus your lupin, your flax, and your sunflowers. There's some other vegetables in there that are included also, but I don't know if they're relevant right in this presentation. In the intermediate group, so those dependent species, they grow best when they have mycorrhizae active and infecting those plants. When we get into the intermediate, there you can see on the left hand side we have our oats and barley, annual ryegrass, safflower, and our wheatgrasses. But on the right hand side, the least of the intermediate group are your rye, your triticale, and your wheat. So if you can, grow those oats and the barley, the ryegrasses, things like that, that helps maintain your mycorrhizae. But if you don't have them, it's not the end of the world. You'll still get a decent crop out of it. So now your non-hosts, now you're starting to deal with your brassicas, your beets, um, lupins. Those are the ones that when we are growing, we have no support for a mycorrhizae. One of the things when we get into regenerative ag, one of the really powerful things that when people are starting to grow canola and they want to continue growing canola and peas together. So now we have something that from the dependent that needs that mycorrhizae. So we're going to put down the peas. They're going to be helping support the mycorrhizae along with that canola, which is a non-host. So that this way, anytime we can, we need to have something growing with that canola to help feed and maintain our mycorrhizae. So building healthier soils. So how do we earn the right to reduce synthetics? How do we build that soil health? And basically, we're going to go through the soil health principles, those five steps, and we want to build and support a mycorrhizae fungi. The cover cropping, intercropping, they check off a lot of those boxes in what we're trying to do. Because when we are looking at our soils, we're dealing with, once again, simplifying things. We're dealing with the biology in our soil, we're dealing with the chemistry in our soil, and we're dealing with some of the physical aspects in the soil. And so in the past, you may have seen something like this where you've seen it where the triangle, the flat part is on the bottom, and they have those three and yada yada yada. In this case, what we're talking about is we're putting the biology on the tipping point, because when our biology is in balance, our chemistry and our physics of our soils are also in balance. When our biology is out of balance, this is when our chemistry, so our nutrient cycles, or our physics, so water infiltration, erosion, things like that, that's when we start having issues. So then how do we support the biology? We support the biology in our soil with soil health. So the living roots, the increasing organic matter, the increased plant diversity, livestock integration, reducing tillage, overall soil health will act as guy wires to keep this triangle from tipping over. And at the end of the day, we want to be building carbon in our soils. The higher the carbon, the less problems we're going to have. Unless we start getting too far and we get into peat soils, which is a new headache. So one of the things when people are asking about cover cropping is, oh yeah, I use a cover crop when I'm seeding forages. And well, in reality, the cover crop that you're putting in for your forages is called a nurse crop, but that's semantics. So the groups that I use for cover crop types is a pre-seed cover crop. So whether it's seeded in the spring and then you seed into it, or you have it over winter. Interseeding, so growing a cash crop with something growing underneath it that's going to terminate during that growing season. We can do a polycrop, growing two or more cash crops together, a relay cover crop, so that you're going to have a cash crop with something growing underneath it, so that when we harvest the cash crop, that relay crop will continue growing into the fall. We can do a post-harvest cover crop, meaning that we harvest our cash crop and then turn around and slap a cover crop in, or we can use a full season cover crop. Each one has its place, each one has its positive, each one has its headaches. Biggest thing when I'm doing these presentations is asking, or when I'm talking to people one on one, is what is your goal? What issues do you want to address? Because if we don't have a goal at the end of the day, how can we measure to say is a success or not? And that's where in the past, when I first got into this, using things like tillage radish, where people would see them in the fall and they would come to me the next year and they say it didn't work. Well, why didn't it work? Well, they don't know, but it didn't work. Now when we start going through and looking at, okay, so now we have it figured out that you're worried about infiltration. Okay, so now we can do a measurement before cedar radishes in the fall. Next year go out and do our infiltration test and say, okay, yes, it worked, or no, it didn't. So now with a proper goal, now we can use the proper species and rates, timing, all of those different things. Now we're going to aim for a more successful cover crop utilization. So when we are looking at goals, where do we start? As a farmer, I hate setting goals in production because Mother Nature is going to be the one that's going to influence the most what whether I'm going to get my yield goals or not. But some of these goal examples are, number one, like the water infiltration, weed suppression. And what kind of weeds? Are we dealing with these nitrate loving weeds, or is it because we have anaerobic conditions? Is it N fixation or phosphate cycling? Is it erosion protection, or is it for animal feed, both for hay and for grazing? All of these are related to increasing the carbon in our soils. So at the end of the day, it's going to be, we're going to be, how do we utilize these cover crops in our management system to get more carbon in the soil? Here we go. This is one of the strategies we can go with. So when we start talking about blends, now we have our goals. This is what we want to do. Now, when we're producing these blends, or we're devising one of these blends, where do we start? If we're looking at uh, those functional plant groups, so when we're dealing with the grasses will produce biomass. It'll has a fibrous root system, so it'll help with aggregating that soil together. But the tons are the one of the keys for the grasses. So it's going to produce that armor. When we deal with legumes claim to fame, that's your nitrogen fixation, and it will add some quality to your cover crop. So some quality organic matter. When we deal with our broadleaves, your brassicas, they're scavengers. I don't use very many brassicas in our system. Number one, because a lot of our farms have canola in rotation or mustard. So adding another brassica, it isn't going to add a lot to the diversity. But it does scavenge. It does have of their fits. I like the brassicas with the tubers. They will accumulate a lot of calcium in our soil, so that there's a definite fit from there. When we start looking at the feed production, they're 85-90% moisture, so they look really impressive in the field, but they shrivel down. They will give you some really good quality. But brassicas to go 5 or 10% of a blend of a full plant stand of a brassica, that's as far as I would ever want to go, unless there was once again special situation where we wanted to go in with a radish or a beet where we really want to bring up a lot of calcium. So your non-brassicas still basically a scavenger, but the positive here is they are mycorrhizal. So with that mycorrhizae, now we can start, building soil. Where with the brassicas, there's uh, there's a little evidence of some mycorrhizal activity, but in my mind, not enough. So using these brassicas, non-brassicas, things like sunflowers, highly mycorrhizal. You have a nice deep taproot. A lot of positives there. And then the last group is the forbs, and that in my mind is one of the missing links. And this is where Dr. Christine Jones, she said in the world the average pasture is 60% forb and 40% grass, which blew my mind because I thought, okay, so where is the legume component? Because when we're doing our blends, we need these legumes. But then on the road home from the meeting, I thought, hey, in a blend, the legumes, when you seed them, they die out after between 5 and 7 years. So that would tell me they are an early successional plant. That early successional plant, assuming we have forbs seeded with it or allow the forbs to establish in there, they're a later successional plant. That is what we're missing in our rotation. In our blends, we do not have any forbs in our agriculture systems right now. So finding these forbs and getting them introduced is one of the keys, I think, in soil health and regenerative ag. So this blend creation now to add this diversity, so we have the grasses, the broadleaf, and the legumes. To add this diversity, now we can start adding annuals, our biennials, perennials. We have warm seasons, we have cool season species. We need to think about where these roots are growing in the soil, when they're growing, when they're their nutrient requirements are during that growing season, and and when they're growing optimally. So those cool seasons, they start growing early in the season. Starts getting warm, their growth rate starts dropping. Now warm season can start kicking in. Then when during the summer, those warm seasons are doing their thing. Then when we start when we start getting into the fall, those warm seasons start slowing down. But then the cool seasons come back in and they start doing their job. These are the things we have to think about. When are we feeding our soil? When we have this vegetative plant that's releasing up to 80% of the carbon back into the soil, and we're growing a monoculture cereal crop, how long is that plant in that vegetative stage? 40 days from seeding until when it starts tillering and starts stem elongation. So that means in a monoculture annual cropping system, we're only feeding our soils for 40 days. So that those other days, we're not. We in that picture that we have up right now, this is a demonstration that Cody Straza from Wood Mountain, Saskatchewan did. He had a barrel that he filled full of soil. He seeded a cover crop on top, and then during his tour, he got a saw and he cut the face off of the barrel, got a garden hose, and started washing the soil away. And this is the root system that is in that barrel that was growing in that soil. An incredible amount of root mass all over the place. Nice demonstration of what is happening below the soil surface. So visualize the blends. I've put together a couple triangles. So we have our grass on the top, we have our legumes on the right, we have our broadleaf on the left, which then I have our brassica, non-brassica, and forbs. With a diagram like this, now we can actually start writing down species beside which group they belong to ensure that we have maximum diversity. It's a quick visualization of how much diversity do we have in our blends. So when we're doing these blends, we want to make sure that we're going to be matching species with our goals. We want to minimize our pest bridges, so we don't want to create a bunch of disease, insect, or grain contamination. What I mean by that is in the United States, they use a lot of fall rye or cereal rye in as their cover crop, which is great because in a lot of cases they go from soy and they're going to go to corn. So to control fall rye isn't a problem. When we start dealing with wheat, barley, oats in our rotation, controlling fall rye in those crops is, well, you just can't. So that can, number one, create some grain contamination in our small grain cereals using fall rye. When we're looking at disease and insects, if we are growing a tight canola crop oscillation between wheat and canola, and you want to throw in a brassica, so a radish or turnip or something like that into that system, I like being 2 years away from canola using radishes. If you don't have 2 years between, it's just going to be an issue with flea beetle. Flea beetle population is going to increase, and potentially some pythium, some rhizoctonium root diseases. So, watching your rotation, minimizing those pest bridges. What you want to do is also, like, part of that is watching that rotational concern. You want to build synergies, not antagonisms. So having a clover in a flax crop that you're going to be seeding canola the year after, that's going to be building nitrogen in the soil so that we are building for that next year's crop. Dr. Christine Jones, one of the things, if you listen to a lot of her work, one of the things she talks about is microbe quorum. And that microbe quorum, what she has found, and it's been repeated by Dr. Zavala in Oyen at the Chinook Applied Research Association, is when we have these cover crops and we have two species in, or we have one species in, it's better than nothing. Okay, great. When we start adding more and more diversity, we have more functional plant groups. What she has found is once we hit between 6 and 9 species of functional plant groups in a mix, this is where those microbe quorum really kicks in. And it, Dr. Zavala has some great pictures that actually Gabe Brown uses in his presentations of monoculture, a crop of triticale, and then I think it was a 9-way species mix of a bunch of different plants. That triticale by itself was sick. It was a really dry year. It didn't look very healthy. Yet right beside it, where she had all this plant diversity, the triticale was thriving. It wasn't a 120 bushel crop, but it was the biomass was significantly higher than just the triticale by itself. And this is where, with those different root exudates, with the different root structures, we have more resilience in our system. And that's that whole idea of what behind Dr. Jones. So as a starting point, your soils may not be the way you want to have them right now, but the question is, how do you want them? How do you want them looking? Where do you want water infiltration? Where do you want, once again, long-term goals? How fast do you want to get there? That's going to be the other influence on how intensive of management are we going to be getting with our regenerative ag management level. And then what challenges or what changes are you willing to make? That's the other thing. When okay, yeah, I want to get into soil health and I want to build soil organic carbon, but I'm going to continue growing wheat and canola. Well, it's going to take a long, long time, and you're going to have to be really stepping outside of the box. When we start wanting to, doing a 5-year, 10-year plan, and we want to do livestock integration, we want to get those 5 main principles of soil health, regenerative ag. We want to get all those 5 in. We're going to start now. Yeah, it's going to happen really quick. The biggest thing in agriculture, well, in the world right now, is a lot of people have lost the art of observation. Observation is a very powerful tool. And that's one of the things that we really have to, when we're doing our day-to-day operation, we need to have a shovel with us. We need to dig. We need to see what those roots are doing. What does their soil look like? What does it smell like? And that's where part of this, I skipped ahead of myself, of these observations. Use your sense of smell. What does it smell like? Does it smell earthy? When you look at that soil, when you dig it up, and not just when you're seeding, you go and look at the top, dig down 30 centimeters and see what your soils look like. What kind of aggregation do you have right at the surface? What do you have 6 inches down? What do you have down at a foot? Do you see any plating in that soil sample? Are you seeing vigorous plant growth, or do you have pockets that aren't as good as what it should be. So you dig in those. Oh, it's because we have a gravel lens there. It's because we have plating. It's because we have salinity. Why are those bad spots bad? Water infiltration. Do you have sitting water on those fields? Whereas your neighbors, there's no sitting water. Or, there's a spot in the field where it was hay 5 years ago. Oh, there's no water sitting there anymore. Oh, cool. What's happening? With that shovel, go out and dig. Do you see earthworms, or do you see evidence that they were there? And what I mean is with the earthworms, if the conditions aren't good, so if it's dry, if it's too hot, if it's not good for the earthworms, they'll drill down, and they could be down 2 feet, 3 feet, and they'll cocoon up. So if they're down that deep, do you want to dig down looking for them? Don't think so. But if you go out and you grab a cube of soil and you flip it over, what you're looking for is little holes. And those little holes are signs of your earthworm moving up and down. So that means they may not be in the top 6 inches of the soil, but they are going to be deeper in the soil, and they have been at the surface in the last year. Once again, depending on tillage and all the other good stuff that you're doing. And the other one is those weeds. Are you dealing with early successional weeds? Or are you dealing with more of a perennial weed issue? And that book I mentioned earlier is those weeds are telling you what's happening in your soils or what's not happening in your soil. And that book is When Weeds Talk by J.L. McCammon. Easy read. In the back, it gives you the soil conditions that plant, what it thrives on. So if that plant is thriving, how do we change the soil conditions so that plant disappears? And it's amazing, once we address those issues, like we can get rid of foxtail within 2 years without doing spraying or excessive tillage. So there are some really neat ways of, once you understand why that plant's growing, how we can get rid of it. So if you're interested in doing some at-home testing, you can do water infiltration test, and if you go to on the web, there's a whole bunch of good resources there. Or go to the LARA Soil Health Lab website. They have some good protocols there. So water infiltration is a good one. Doing a slake test, and this is what the picture is I've taken 2 clumps of soil that I've air dried for a week, and then you put them into a coarse mesh, and you put that in water, and you can see the soil particle on the left. It's still intact after 3 hours, whereas the one on the right has completely disintegrated. So the one on the left, we have really good soil aggregation. That soil is holding together under these super wet conditions, where the one on the right is more bacterial soil, and it just disintegrated. It doesn't have that good soil aggregation. If you get a penetrometer, you can see how hard your soils are and where your hard pans are. You can deal with a micro aggregate stability. So similar to this lake test, where you would put it through a colander, so you get 2 millimeter samples of soil micro aggregates, and you put that in water and let it sit for 15 minutes and see how much of those aggregates are still intact after 15 minutes of sitting in water. So the longer the better. The key is make sure you're not looking at rocks, because rocks would stay in water a long time. You can do bulk density of your soil. So taking a known volume of soil, drying it down, and finding out how much that weighs. And that's an indication of how much airspace you have in your soils. So if you have really tight soils with low airspace, you're going to have a really high number. If you have good organic matter, you have good aeration, good pore spaces, you're going to have a relatively low bulk density, which is what we want. There's a trend a couple years ago where soil your underwear. So grab a pair of cotton underwear, and once again, cotton socks, whatever, just a piece of cotton, bury it in the soil, let it sit for 2 weeks, 4 weeks, 8 weeks, whatever duration you want. Dig it up and see how much of that cotton has broken down, and a quick indication of what kind of soil biology activity you have. A little more technical is the tea bag test. So you can take black tea and green tea in bags, weigh them, put them in the soil, bring them out, do a weigh after, and you get once again a real good indication of what kind of soil biology activity you have. Or you want to go to the full meal deal. This will cost money, but the information that Dr. Zavala can give you on your soils is really remarkable. Not only will she give you the test results and some consultation with it, if you talk to her and line it up, you can grab your soil sample, send it in, and find out when she's going to be doing it, and she will, in this COVID world, there's ways around it, uh, and I don't know. Once again, phoning ahead is the key. She may be able to take you through and show you what your soil samples look like, show you what kind of nematodes you have, what kind of bacteria. This whole, give you the full meal deal of what's happening and how she does her tests. Now, a couple examples of what some of these soil samples look like and what are we looking for. Here's an example of two soil samples that I've taken about 20 meters apart. On the left is the farmed land, and you can see that there's not much for soil structure. It's dry. It's a dark, or it's a very light brown. And guess what? There wasn't much for plant production, plant growth on it. On the right hand side, I went over to the field margin into the where the brome grass was growing. There was trees along the fence line, and you can see that it actually there was moisture in it. This is on a fairly dry year, but there was actually moisture in it. You can see how the soil, it looks like ground fried ground beef. It has a good soil aggregation, and guess what? The plants there were just about a meter high, whereas the thistles on the ag land were 5-6 centimeters high. So not productive. Whereas the natural system, the soils were functioning, they were active, things were looking good. Here's another field. This is a cornfield, and not a lot of not a huge amount of diversity. It's conventionally farmed, and you can see in the top couple inches, you can see that there was some plating in there. Not a lot of soil aggregation, and you can see that where this blue line is underneath it, it looks like just a block of cement. And so in a field like this, we're going to have infiltration issues. We're going to have anaerobic conditions. We're going to have a lot of issues. Below that blue line, you see a few small roots that have made it through and are getting in there. But in this case, we're going to need to use cover crops with deep tap roots. We need to wake these soils up. And that's one of the things that we're looking at. So these 6 steps to significant change, and if you're listening to this presentation, you've already experienced a need for change. You're diagnosing the system, you're pioneering tactics, and this is where, as we go along, we're going to repeat this, so that we're pioneering practices, we're enabling the tipping point, we've sustained the transition. Now we set the new rules for the new mainstream. When we get to that, guess what? Now we're going to see more need to change. We need to re-diagnose the system. We need to pioneer new practices. And so it's the next level. It's that next step. And it's not a bad thing. There's going to be some times when we diagnose the system and we may misdiagnose it. And so, we see these thistles. So, okay, we're going to have to go in deep rip. Okay, when we go in deep rip, okay, so we've created new issues, so it's going to be more bacterial in the future. It's going to collapse on itself, and the thistles come in thicker if we miss that window. So these are the things that we always have to be looking at. So we're going to be setting new rules. Now we're going to be looking at, okay, so now what do we do next? Sir Albert Howard was an agronomist. He's the godfather of the modern organic principles, production principles, and he wrote a book, The Agriculture Testament. Wonderful reading from the early 1900s. And the scary thing is what he was complaining about, they're still going on today. And one of the quotes that I love from his one of his books is the health of the soil, the plant, and the animal and man is one and indivisible. And when you understand these ecosystems and how we're managing, it makes so much sense. And so some of these issues that we're seeing today, things are falling apart, is because we have to start go back building our soils. The other really great quote that I love when we start getting to regenerative ag, and you're gonna if you're just starting into it and you don't have a good support team and you feel you're all alone, don't worry about criticism from people that you wouldn't seek advice from. So that when the neighbors are laughing because you have, your weeds, your early successional weeds when you're starting to convert fields over, and believe me, I've lived it this past year. There's one of our neighbors reported me to the weed inspector because of the weeds. Well, it turned out to be chicory that they didn't know what it was, and they got excited about it. But anyways, the whole thing about this criticism is make sure you have this good mentorship. Find a mentor, talk to them. Uh, Attend crop tours, or go for one-on-one tours to someone that you respect, that's been on this road. Take a look, see what they're doing, what are they seeing, how they doing it, because there isn't a easy button on this. It's going to be individual, so that as we go through, you're going to pick out the information of what we've talked about in this presentation. You're going to have reading books from Nicole Masters. You're going to listen to podcasts. You're going to listen to all this stuff, and then you're going to have to try and figure out how you're going to integrate it into your system. And that is the challenge. And talking to people and throwing ideas, throwing stuff against the wall and see what sticks. This is that whole part. So don't worry about the criticism, but go out and find someone that actually is living it. And that's one of the keys of moving this forward. What I love about regenerative ag is the people that are involved in it are open to sharing. And this whole sharing, and you want to hear about the mistakes on my farm, I'll tell you about my mistakes, and there's enough of them. But I don't see them as mistakes, I see them as learning opportunities. So thank you for your attention. If there's any questions, you feel free to get a hold of me. You can get a hold of me at on Twitter and Facebook also. And good luck on your journey along regenerative agriculture.